The Cannabis Conversation. A European perspective on the emerging legal cannabis industry.
Welcome to the Cannabis Conversation with Anuj Desai, where we explore the new legal cannabis industry by speaking to the professionals that are helping to shape it. On this week's episode, I am beyond honoured to speak to Professor Raphael Mechoulam. Professor Mechoulam has had a huge impact on our understanding of the plant with his groundbreaking research and has paved the way for lots more research to come in the future. Remarkably, he's turning 90 soon and is still working hard on discovering more, which is a true inspiration for all of us. I feel very blessed and grateful for the opportunity to speak to the great man himself. I'd just like to say a quick thanks to Zofie Postin from Technion University who helped set this up for me. I hope you enjoy the show. I'm very excited to have Professor Raphael Mechoulam on today's show. For the few of you who don't know who he is, he's commonly known as the Godfather of Cannabis research and has done many, many amazing things within the medical cannabis sphere. Amongst many things, he was the first to isolate THC. He discovered the structure of CBD, and he also isolated the endocannabinoids and andamine and 2-AG. He's a professor of medical chemistry at Hebrew University of Jerusalem and is still active in researching cannabis to this day. Professor Mechoulam, welcome.
Thank you. Thank you for inviting me.
No, real pleasure, real honor, actually. I'm really excited about this. There's so much to ask you, but, usually I start the show by sort of asking for your background, but you've got such an amazing background that probably most people know about. Maybe we could, if we take it back to the very beginning of your relationship with cannabis as a researcher, how did it all start for you?
Well, I'm a chemist, and I'm interested in the chemistry of natural products. And I was surprised to find out that while morphine had been isolated from opium in the early 19th century, and cocaine had been isolated from coca leaves, again, 19th century, the active compound or compounds of cannabis had never been isolated in pure form. Some very good research groups had worked on that, but for some strange reason, the active compound had never been isolated in pure form, and its structure was unknown. And the structure of cannabidiol, which had been isolated, was unknown, and it was not really realized whether we are dealing with a whole mixture of compounds or not. So as a chemist working on natural products many years ago, one of the projects I started on was the chemistry of cannabis. And I got about 5 kilos of cannabis from the police. They didn't make any problems. Probably today it would have been much more difficult, but at that time, the police just asked whether I'm reliable. They asked the head of the institute I worked at, and the head of the institute said, of course, he's reliable, although he had never met me before, and went over and took 5 kilos, and together with colleagues started work on the chemistry of cannabinoids. And our first project was the isolation again of cannabidiol, and we isolated, and using modern methods we were able to elucidate the structure. Then we went to THC, and again we isolated, I believe for the first time in pure form, THC, and we also together with colleagues looked at the activity of all the cannabinoids which were isolated, about a dozen cannabinoids, most of them unknown, and the only compound which showed psychoactivity in monkeys turned out to be THC. And we, at that time, also elucidated the structure, synthesized it, and it became available to scientists for research. At the beginning, nobody was particularly interested. As a matter of fact, when I asked for a grant from NIH, the US NIH, which at that time was quite open to researchers abroad, not only in the US, they said, well, nobody uses cannabis in the States, or marijuana. It's only used in Mexico. So when you have something more relevant to the US, ask us. Well, it turned out that within a year or two, cannabis became relevant to the US for some strange reason. And people from NIH came over, and asked me to please continue. And they took the world supply of THC, which we had. They smuggled it back into the US, assumed that they didn't have okay. And some of the research that was done in the US was done with the material we gave them. So one of the major projects my lab continued to work on was cannabis, originally mostly chemistry. So we isolated cannabigerol and some of the cannabinoid acids and looked at their structures and looked at their activity. So we went into pharmacology, and quite a few of the compounds, almost all the compounds we isolated were new. And so after working on the pharmacology and started clinical trials. So that was in the early 70s, early 80s. And we went on working in all these areas. Then I thought that we should try to find out the mechanism of cannabinoid research. And there was some mistakes in the literature. I wouldn't go into the details because they're technical, but most people didn't believe that there is a receptor or there are compounds that will act on these receptors, which was wrong. And indeed, in the US, a young researcher found that there is a receptor. And so if there is a receptor, obviously it's not because there is a plant out there which makes a compound that binds to these receptors, but these receptors are in the brain or in the body in order to be activated or suppressed by compounds that the animal body makes, that we make. So we went ahead looking for these compounds. It was quite a problem because these compounds are found in very, very small amounts. But ultimately, after a few years of research, and had postdocs working on that, and some of my PhD students, we isolated two compounds. The first one we called anandamide, on the Sanskrit name of Ananda, which is happiness. We thought, and I still believe, that these compounds are involved with emotions, and Ananda in the Sanskrit is happiness, extreme happiness. Another one was 2-AG, which is the short name for a very long chemical name. So these two compounds were investigated by us and by many others. As a matter of fact, the anandamide has become a very, very interesting problem for many years, for many researchers. The paper that we published, 1992, has been cited almost 6,000 times, and people believe that it is involved in essentially almost all human diseases. This was something reported, published, stated by people at the NIH in the US, said the endocannabinoid system is involved in almost all human diseases. Now, what is the endocannabinoid system? There is the receptor. There are two receptors, as a matter of fact. There are the two receptors. There's anandamide and 2-AG, and enzymes that synthesize these compounds and the enzymes that break down these compounds. So all this represents the endocannabinoid system. And indeed, it has been found to be involved in a huge, huge number of physiological reactions, in changes that happen in our body, in disease or in the normal state. And this has brought the interest of many, many people. After all, the anandamide and 2-AG are essentially neurotransmitters. And for all I know, they could have been discovered together with the well-known neurotransmitters that were found in the 30s, so many years ago. But they were not, because the cannabinoid system was not known. So there has been a lot of work on these endogenous cannabinoids, endocannabinoids. So when people speak of the cannabinoids, they mostly think of the plant. Well, the plant is one phase, important, but one phase, the cannabinoid knowledge, cannabinoid science. The second phase are the endogenous cannabinoids, namely anandamide and 2-AG. And now we are involved in the third phase of cannabinoid research. You see, anandamide is a relatively simple compound, relatively. It is made from a fatty acid and an amino acid. And our body, animal body, has about 20 fatty acids, 20 amino acids. So by using the same mechanism for synthesizing anandamide, it can make a lot of other compounds. And indeed, the animal body does that. So we have in our body about, 50, 60, maybe more of these anandamide-like compounds. Now, the body doesn't make things because it has nothing to do. It makes them for some reason. So we have been working on these compounds for the last 10, 15 years, and this represents the third stage of cannabinoid research. We have been working on them, and they are of extreme importance, these anandamide-like compounds. One of them, or maybe a few of them, help the brain when it undergoes brain trauma. If we compare a brain that has received these compounds and a brain that has not, the brain that has received these compounds shows much less brain trauma. So these are compounds, these anandamide-like compounds are compounds that protect us in the brain and as well as other wounds. Then with colleagues, and all this is done with colleagues. You see, as I said at the beginning, I'm a chemist, but I work with a lot of friends and colleagues all over the world, whether it's in Brazil, where we worked on epilepsy, or in the US, where I work now on a few other projects, in Canada, and I'll tell you a bit about it in a second, and in Italy and so on. So it is not possible for one lab to do all these things because they involve chemistry, pharmacology, biological sciences, clinical things, and so on. So if one wants to see the whole picture, one has to work in collaboration. So we used to meet, now because of the corona, we cannot meet so often, we used to meet at meetings, drink a lot of coffee together, sit around the table and discuss the possibility of a certain research. So we, as I said, looked at brain trauma. Then we were working on bones, and we found that the bones after the age of 50 or 60 in women start producing a compound, again, an anandamide-like compound. I wouldn't go into the chemical details, of course, which blocks osteoporosis. Women after the age of 50, 55, all of them should have osteoporosis. They don't. And the reason, one of the reasons is they make an anandamide-like compound which blocks osteoporosis. So that's part of the cannabinoid system. And quite a few other things. And I'll just end by telling you about what we're doing at the moment, again, in collaboration with a group in Italy, a group in the US, a group in Canada. And we are looking at compounds that are involved in addiction. You see, addiction, people used to think it's something just in the brain, it has nothing to do with biology. Well, no, addiction is a disease. And our body, our brain, believes that if there is a disease, it fights a disease. So we thought, let's find out whether our body, our brain, makes anything that fights the disease. So we have been working on that for several years, and our 4 groups found that part of the brain produces a compound which blocks nicotine addiction. It's not always successful, but there are people that get addicted to nicotine and people that do not get addicted to nicotine. Most of the research, of course, has been done on those that are addicted. Probably it would have been more profitable, scientifically more profitable, to look at the people that don't get addicted. Why don't they get addicted? Anyway, so we found a compound which blocks nicotine addiction. And this has been going on in collaboration with my colleagues and my friends. And then we looked whether this same compound blocks opiate addiction. No, we're sorry to say it does not. But this particular compound, oleal glycine, we have published all that. It's a mixture, it's a molecule from oleic acid and glycine, 2 well-known compounds in the body. Oleal glycine blocks the withdrawal symptoms not only of nicotine addiction, but also of opiate addiction, in mice at least. And that's important because many of the opiate addicts cannot leave opiates because they're afraid of the withdrawal symptoms. And it is a major, major problem. In the US last year, about 60,000 people died of overuse of opiates. So here is a compound that possibly can be used as in opiate research. So we started with the plant, and we are working now on withdrawal symptoms of opiates with compounds that are cannabinoid-like compounds. So we have 3 phases of research: the plant, the endogenous cannabinoids, and endogenous-like compounds. Well, here is in a nutshell what I've been doing for the last few decades in this particular area.
Yeah. Wow. So much there. It's incredible. And actually, the idea of collaboration is one that I wanted to ask you a couple of questions about. But, while we're here, some stuff just came up. So currently, there are 2 endocannabinoids that you've named, the 2-AG and anandamide. Would you class these other anandamide-like compounds as endocannabinoids? Or do you think there will be more than 2 endocannabinoids?
Well, giving names to things doesn't matter. It's what's important, what do they do? Their structure is like anandamide. Anandamide is formed from a fatty acid and a derivative of an amino acid. These compounds are the same, a fatty acid bound to an amino acid or a derivative. So if one wants to call them endocannabinoid-like compounds, well, the person is welcome. If one doesn't want to call them that, it doesn't matter. Science is not about terminology. Science is about what compounds do. So there we are. Yes, there are additional compounds. People have found in Italy 2 or 3 additional endocannabinoids were found. They have not been researched very thoroughly, but yes, they are there, and I believe that they may turn out to be important. But at the moment, most research, essentially all research, has been done on anandamide and on 2-AG, and we know quite a lot about them, how they are made and what are the enzymes and what they do. Basically, anandamide, for example, is present in very, very low amounts in the body. It is formed when and where needed. And then it is a strange one because most compounds of this type are formed in the nerve. The nerves are not just a wire from the foot to the brain. They are cut, and there is a space between part of the nerve and the other part of the nerve that's called the synapse. And most of the synapse goes in the ongoing part of the synapse and the presynapse. So these compounds are made in the postsynapse when needed, not anytime. When needed, then they move to the presynapse, and there is the receptor, and they bind to the receptor, and they do all kinds of things. For example, they regulate other neurotransmitters. So it is a compound of major interest.
And do you think it's possible that there may be more receptors as well within the endocannabinoid system?
I doubt whether there are more receptors. People have looked into that, but these are lipids, and lipids bind to a certain extent to others. So yes, they bind to others, but it seems that the major the major effects are by binding to their specific receptors. The receptors are cannabinoid receptor number 1 and cannabinoid receptor number 2. Now, but of course there is a difference. Cannabinoid receptor number 1, when stimulated, causes all the marijuana-like effects. Cannabinoid receptor 2 is different, completely different. It does not cause these things, but it is a protective one. So if it is a protective one, its stimulation blocks all kinds of disease states. As a matter of fact, a long list of disease states associated with the CB2 receptor. So we have been working, for example, on compounds that bind only to the CB2 receptor. And these compounds do not cause, from a medical point of view at least, undesirable psychiatric effects or psychological effects, but they are great interest in lowering inflammation, in doing all kinds of other things. So chances are that this type of compounds that bind specifically to the CB2 receptor, will become drugs. And I'm aware of at least one major company that's working on a CB2 specific compound. And we have also, together with companies, also published compounds that are CB2 specific, bind only to the CB2. So maybe we shall have cannabinoid drugs that are not THC, but bind to the CB2 receptor alone. Let's see. You'll be seeing it. I won't. I will be 90 in about 2 months.
Wow.
So if they come out in a few years, chances are that I'll be looking at them from somewhere above.
Well, I hope not. But yeah, look, I mean, it's amazing there to break down the explanation. One of the things I really liked about what you said, it's almost like the body produces things to deal with certain ailments or conditions, but you supplement things from externally, i.e. the plant sometimes, if your body can't create those things themselves. So that sounds like a fascinating area.
Well, yeah, that's the normal state. I mean, the body fights disease and we try to help it. So the body, for example, has an immune system and it fights disease. But if we can help the immune system and enhance the activity of the immune system, then probably we're in a better state. So here we have the same situation. The body produces compounds that are effective. So if we synthesize these compounds, and we have synthesized many of them, we synthesize the compound and add a compound to the injured animal, we see better results. As I mentioned brain trauma. Now the brain tries to, if we cause a minor brain trauma, the brain tries to cure it. But if we give one of the compounds, in this particular case 2-AG, synthetic 2-AG, the brain works much better. As a matter of fact, after brain trauma, 2-AG goes up. Its concentration goes up because the body tries to fight the disease. Mm-hmm. Well, we can help it. So we give a little bit more. And yes, it's much better when we give a lot more of this compound. So here we are. This is actually not very different in many other disease states. The body tries to lower the disease damage, and drugs are in there in order to help it.
Yeah.
There we are.
Yeah. Makes complete sense. So I'd like to pick up on a couple of things that you talked about. And one of the major things is around collaboration. And you talked about you've worked with scientists across the world. Who are the people that you're working with at the moment? Who are the people that you're inspired by in terms of research in this field?
Well, it depends. I mean, if we're working in a certain field, I understand the chemistry of the cannabinoids and the metabolism of the cannabinoids. And while I know a bit of pharmacology, I'm not a pharmacologist. I don't know how to do that. So there are pharmacologists that the opposite side, they know pharmacology, know how to do experiments in pharmacology. They may know a little bit less of the chemistry. So it seems quite normal that we should sit together, drink a lot of coffee, discuss things that are common. And as I believe I understand some pharmacology, and they understand some chemistry, and if we have additional people around, so we look at the major problems that involved. To give you an example, I mentioned our recent work on addiction. I'll give you an example of work that we did, about 40 years ago, in epilepsy. It was known historically that cannabis had been used in epilepsy. As a matter of fact, a friend of mine in Yale, a historian, had found some papers in Paris in which it was said that an important person in the Muslim world in the 15th century had epilepsy, and he was given cannabis, and he had to take the cannabis throughout his life in order not to have the epileptic attacks. So it was known. So we went ahead. How did we go ahead? Had a good friend, Professor Carlini in Sao Paulo in Brazil. And Professor Carlini and I, pretty good friends, and we decided, well, he will look at the epileptic properties of cannabidiol, and we'll prepare a lot of cannabidiol from hashish. Now, there was no cannabidiol available at that time. It's not like today. So we got kilograms of hashish from the police, and we isolated almost half a kilo of cannabidiol, quite a lot of work by my students. We isolated almost half a kilo of cannabidiol and started sending to Carlini in Sao Paulo. And Carlini, after work on animals, tried it in 15 patients. And yes, half of them were placebo, namely they didn't take anything, and the half were given cannabidiol. And indeed, those that were given cannabidiol, about half of them didn't have any attacks, or almost no attacks, for 3 months. And part of them had very few attacks, and only one was not affected. So here, this is the type of research that we do. I mean, we sit down together, we discuss a problem, we find out what's the main thing that has to be discovered. And then in the lab, my lab does the chemistry, the other lab, in this particular case, does the pharmacology and clinical science. And ultimately, we see the results and publish them together. This is what we have been doing. And I like working with friends in the UK. As you're in the UK, I've been doing a lot of work with Professor Pertwee. He's now in Scotland. He used to be in Oxford, and he did his PhD there. And I also worked with his boss, Professor Payton, at Oxford, and we also published quite a bit on a long list of compounds. He's a pharmacologist, and he's an outstanding pharmacologist, and we've been working together for many years.
Great.
We are.
Fantastic. And so obviously, as you said, you've been researching for many decades. How do you think the internet has changed sort of scientific research? Because now you can obviously collaborate with someone in South America much more easily than you could do in the 60s.
Well, I'm not sure that now it's easier. In the 60s, very few people were interested, but no, we did. There is no problem with the internet and the computer, and there is no problem working with people with friends and colleagues all over the world. I see no reason why not. At the moment, for example, quite a bit of our work on addiction is done with a friend in Canada, and she works in her lab is around Ontario, but she's at present in Western Canada, which is a small place. Canada. It just takes 3 or 4 hours to fly from where she is to her lab.
Yeah, that's great. That's great. Yeah, I mean, it's brilliant that everyone can speak to each other much easier now and share ideas. Of all of the things that you've done, where do you, and this is a really hard question, but what's one of the things that you're most proud of, piece of research that you've uncovered?
Well, I would think that discovery of the anandamide is of some importance. This it helped helped us find a new biological system, a major biological system in the body, which one doesn't find every day. And I think that this is of major importance now. But I can think of THC, the discovery or the isolation of THC from the plant. But I think that the anandamide story is more important because it has taught us of what we are and what we do, and all kinds of explains our emotional states, and chances are that it may even explain our personality. You see, there are about 8 billion people on this planet, and I understand that all of us are different. Now, how come that 8 billion people are different? Well, it's quite possible that they are different because they have there is a long list of cannabinoids, and this long list of cannabinoids can vary to a huge extent. A little bit more of compound A, a little bit more of compound B, and if you have 60 or 70 compounds of this type, you can have any number of people there. And we actually published that a few years ago. Hope that somebody will have the ability to go ahead and prove that.
Yeah, that sounds like a very big task. Well, it actually kind of fits quite well into the next question that I was going to ask. And with a plant as complex as cannabis, or any other plant that's very complex, how do you fit it into the modern Western scientific kind of school of thought?
Well, chemistry has been around for almost 200 years now, and working on plants has developed. At the beginning, it was extremely difficult. Now it's a little bit less difficult because of the modern methods for proving the structure of compounds and isolating them and synthesizing them. But I see no major difference of what people did 100 years ago with their methods, which were not as good as today's methods, but the same way of thinking. Let's isolate the compounds and see what they are. Now, with cannabinoids, it's a little bit more complicated because of an effect we call the entourage effect. And it's the entourage effect is that the effects of both plant compounds and some of the endogenous compounds, the effects also depend on additional compounds. For example, extracts of cannabis sometimes work better for patients than the pure THC or the pure cannabidiol, as a matter of fact. So they affect the metabolism, or they affect the compound in some other ways. And this entourage effect has not been well investigated, but it does seem to be there, not for all the effects of cannabinoids, but at least for some of the effects. So there is a lot of work to be done, particularly on the entourage. And this is the preclinical part. In the clinical part, I'm sorry to say that there hasn't been enough work. People use cannabis for a lot of diseases, but the clinical background is not there. The clinical background is in, let's say, in epilepsy. We did initial work 40 years ago, 30 years ago. It was repeated 2 years ago on a large scale in the US. And now it has cannabidiol, essentially pure cannabidiol, has been given okay by the Food and Drug Administration in the US. So for epilepsy, that's fine, but not for anything else. Schizophrenia, excellent papers by a German group, by other groups, showing that cannabidiol seems to be an excellent drug in schizophrenia. But although the German group has published an excellent paper, and other groups have done so too, nobody going ahead with that. Or diabetes type 1. It is a disease that needs new drugs, and we have shown in animals that have the same mechanism for diabetes type 1 that cannabidiol is extremely useful, but we don't have a clinical trial. So maybe people use it for diabetes type 1, but this is not the way to do it. It has to undergo clinical trials. And we have the same for a huge number of disease states. So it's a shame that quite a bit of the clinical work is not there. So people are using cannabis the way they used cannabis. They could have used cannabis 200 years ago. My neighbor said it's okay so I will use it. Well, this is not what should be done.
Yeah. I mean, I guess one of the challenges is just the number of different compounds that you have. Obviously, you've got the cannabinoids, but you've also got terpenes and other things that are working. And how do you know which ratio does, when you've got so many different inputs? Is that part of the problem?
Yeah. Well, what I was saying is that it's not that your point has not been researched. Even with the pure compound, there hasn't been enough work.
Right.
I mean, we know that cannabidiol works in anxiety, but I'm looking forward to see. Well, anxiety is a complicated system. Not all anxieties are the same. I want to see clinical work on anxiety. Want to see clinical work on depression, different types of inflammation, and so on and so forth. I can give you a list of 20 or 30 disease states that most definitely people should be looking at cannabinoids because they may be active, but until clinical trials have been done, I'm sorry, one cannot put cannabinoids in the list of approved compounds.
Yeah.
We need clinical trials.
Yeah. I think that's a recurring theme that we keep hearing. Hopefully it will be accelerated soon. So cool. As we're getting towards the end of the interview, maybe you could talk a bit about what you're doing now. I know you're looking at THCA and CBDA. Would you mind talking a bit about that?
As I said, we're working now on addiction. In the chemical side, the plant doesn't produce cannabidiol or THC. It makes the precursors of cannabidiol and THC. It makes something we call the THC acid and the CBD acid. These are the compounds that the plant makes. And they slowly convert into THC and CBD. They're not stable. As they're not stable, there hasn't been much research on them. And I thought that that's a mistake because they may be very active, and there had been papers showing that. So we first stabilized chemically. We stabilized THC-A and CBD-A, simple chemistry. And now we could look at them, and we found out in collaboration with colleagues. The first paper was with Canadian colleagues and Professor Pertwee in Scotland, and we found that, yes, it definitely acts on anxiety. And it also, by work in Canada, been shown to have many other effects. For example, these compounds lower vomiting when people get the anti-cancer chemotherapy. Very frequently they had side effects and they start vomiting and so on. These compounds in mice block that type of effect. So chances are that these compounds will be as important as CBD and THC. So we're working on the cannabidiol acid as a hopefully important compound. I wouldn't say new compound because cannabidiol acid has been known. We reported it way back in the 60s. At that time also a Czech group was working on that, and they also reported it in the 60s, but there hasn't been much work done since then. So I believe that one should be looking at these compounds, and maybe they have all kinds of additional effects. So we have THC acid, CBD acid, cannabigerol acid. We discovered cannabigerol again in the 60s and cannabigerolic acid in the 60s, but not much work has been done on these two compounds. So maybe, and so we're working one of the topics. My group is very small. I never had more than 2 or 3 people working with me. So there we are.
Yeah. Wow, that's really exciting in the area of addiction and the various other things that you're looking at in relation to the acid. So I look forward to seeing how that all goes. I'll leave you now because you've been very generous with your time. Thank you so much, Professor Mechoulam, for joining me today.
It was a pleasure. And I hope that, more work will be done on those topics that I mentioned, particularly on the clinical side. It was a pleasure.
Thank you, Professor. Have a good day.
Same to you.
Thank you. Thanks for joining me for that. Hope you enjoyed the show. If you did enjoy it, please hit subscribe on whichever podcast platform you use. I'd also love a nice 5-star review on iTunes if you have the time. You can also follow the Cannabis Conversation page on LinkedIn and also sign up to my mailing list via cannabis-conversation.com. Until next week, have a good one.